Today we're going to talk about the biostatistical rigor and transparency in biomedical research. I'm sure, a lot of you took the biostatistics, and whenever you hear the word of biostatistics, and you get to frown, and what do I get to do with it, and that anxiety kicks in, right? So I'm trying to remove that barrier a little bit today, and then talk about the rigor and reproducibility criteria that came in from the NIH. So NIH and a lot of other agencies, the problem came in that they were not able to translate and replicate results. That became a big problem, including the journals. Nowadays, they want the actual data set and the coding of the software that you've done, because they were not able to reproduce the results. And some of these slides are borrowed from the SIU medicine, and they had the training series on the rigor and reproducibility. And there are a lot of documentations that are produced by the NIH. NIH plans to enhance the reproducibility, so whatever you write it, we'll be able to duplicate in the lab, from lab A to lab B, or from Birmingham to Chicago and any other place around the world, around the country and world. And then the balances sex in cell and animal studies, right? And when you do an animal study, some of you know the certain gender does respond better than the other gender. So, as an overall, unless you are doing research on the ovarian cancer or prostate cancer, we want to see the combined animal study, and then you can talk about the trends of the gender. So those are some of the things that we're going to talk about as well. So NIH wanted to have to see a methodological rigor in study design in order to translate results. And ultimate goal is translate basic science into clinical studies and human intervention. And then, if there's unsuccessful clinical trials, it expose the patient or study patient to harm. And it also wastes a lot of resources that come out to get put out together. And NIH came up with the 4 areas of clarification in the NIH grants. Those are scientific premise, and which translate into a scientific background, scientific rigor, and relevant biological variables, and authenticity of key biologics and chemical resources. Today we're going to focus on the scientific rigor and biological variables. So those guys who's working in the lab, do any of you guys use the randomization? That's great. Some lab, I've seen it happening. The batch who's living in the animal cage down here, because of the ease of treating the animals, that happens. Randomization is very important, the principle to reduce the bias. And then also the blinding. Have you guys heard about the blinding? So whoever made the treatment, right, if it is the same person who's measuring the thing, that can also produce a bias. So if technician A does the treatment, the one who's blind to the experiment, if they read the experimental outcome, that may also reduce his bias. Because I did it, I tend to like to show the more effect of these drugs or treatment, right? And also doing the sample size calculation. You cannot add ad hoc, additional number of animals. At the end of the 10 animals, you reach the 0.06 or 0.07 p-value. Let's just add a few more. That's big no-no. You have to do the prior, sample size justification, and then also the proper data handling. You cannot throw one outlier, because it happened because of the treatment. The number is too big or too small. You cannot just throw an outlier. You need to have those things documented before you go into the trial or study. So in the grant application and grant review process, scientific rigor and consideration of the biological variables such as sex falls into the research strategy section. And it gets scored as an approach, and then also as well into the contribution to the overall impact. So for the scientific rigors, we have to describe the experimental design and methods proposed and how you guys will achieve robust and unbiased results. I don't know whether each lab has a protocol written up. Having a concise protocol to follow, we're going to produce a lot of unbiased results, and it will be a robust, well-controlled experiment, and also be able to reproduce under well-controlled conditions using reported experimental details. More detailed information that you have, it's better off. I mean, if you did the experiment at certain time, certain day, I mean, that also may contribute to the bias too. So as we discussed, using the independent blind assessment is a good approach to reduce the bias. And then selecting a primary endpoint prospectively, not at the end of the trial, not end of the study. You measure like 20, 30, 40 measurements, and then whatever gives you a p-value less than 0.05, you want to use it as that one as a going forward. That's not a good idea. You need to decide what will be the primary outcome and do a proper sample size justification. Other things you may explore and see for the next study, but not as this not as the primary outcome for the current one. And then randomization is very important principle. And then define inclusion and exclusion criteria in advance. And we're going to look at the example later on, and also predetermine how to handle the outliers. I mean, don't just throw the data out when you observe it. You need to define it before you go in. And conduct interim data analysis that may be feasible, redo the sample size, redo the sample size calculation. So here's a brief video from the NIH. They have a lot of training videos that's available, and this tells you about having a proper protocol and then detailed protocol. Let's watch this video together for quick.
I performed the experiment exactly as Donna described in her protocol, and compared with her results, mine just aren't matching up. I just can't get the results to be statistically significant.
Well, something may be wrong with the way you're doing it then.
I guess.
This was the 4th protocol that Donna perfected when she was postdocing here in the lab. I mean, this is a good study design. I really don't get why you can't reproduce it either.
I'm following everything to the T exactly as described. It's just so weird that it won't work.
Well, let me ask you this. How many samples did you use?
I used 8 mice per group.
Let's try this. Add 2 more mice to each group. Then try a different statistical method. If I remember correctly, I believe Donna performed a 2-way ANOVA. See if changing those parameters gets you the results we're looking for here.
That's a good idea. Remember, Robin, toss out anything greater than 10. The normal range for living cells is 3 to 7, so anything higher than that is probably due to cell death. So keep that in mind. It could make a difference.
Okay. Thanks, Jamal.
And let me know how you do, okay?
Of course, Dr. Fielding.
Please call me Harry.
Dr. Fielding.
Hey, she's new. You did the same thing.
No, I didn't.
Yes, you did.
Oh, come on.
Yes, that's whatever.
So, where are we here?
I did what you said, and I added the 2 mice per group, and look, it's still not statistically significant. These results are inconclusive, and they don't match with Donna's results at all.
And you varied the statistical test? Ran a 2-way ANOVA this time?
Yes. I mean, why is this happening?
Robin, I've been watching you a little bit in the lab here, and I hate to say this, but it seems like you're struggling a bit.
What?
Sometimes students come in here and just can't hack it in grad school.
It's just embarrassing that I can't repeat this test.
Well, I'm worried too. We do consistently high-quality work here, Robin. We just can't afford to lower our standards like this.
I'm sorry, Dr. Fielding.
Okay. Just bring up Donna's.
God, here he comes. Hope I don't get another lecture.
No, you're okay. Okay?
Jamal and Robin, do you have a second?
Of course.
Sure.
Well, I think you both know that I've been concerned about Robin's work and that experiment not being reproducible. So I decided to take a look at Donna's old lab notebook. Took a while to decode, but I think it was worth it. I found something.
Really?
First, I found that some samples were excluded from the experiment, but there's no explanation as to why. There's no justification for leaving these out, and no criteria for outliers or excluding samples appears anywhere either.
I wonder why.
So if she'd included those samples in the analysis, what would it have looked like? Is it similar to what we were looking at before, or is it more like Robin's results?
More like Robin's.
Wow. Really?
Mm-hmm. And there's more. As I was digging further into the notes, I realized that all of the outliers were actually samples from female mice.
That's so strange.
And significant. Rigorous research, which, by the way, Robin, you were doing, requires considering sex as a biological variable when using mice, or any animal samples. I was the one who was not doing a very good job, and I should have caught that.
That's okay.
No, it isn't. I never even thought to tell you which sex to choose when I told you to add those 2 mice samples.
Well, I didn't think of it either.
Yeah, but I'm the PI, and I'm supposed to train you in designing and conducting experiments. And I changed the design of the study halfway through. I had no business doing that. So what I'm thinking is, I should take my own medicine, so to speak, and get back to a more carefully designed study. More consistent, and most importantly, much more robust. I think we should stop, regroup, and start over.
So if you go to that website, I mean, there's 11 more videos that's available. It's entertaining, and then but it hits the key point of doing the lab research. It'll be very beneficial for lab person who's working in the lab to go and visit and go through the videos available at the NIH website. So in the research strategy, right, we have to succinctly state what is planned. That includes the sample numbers and blinding that you plan to do, and statistical power and analysis, and describe the experimental animal numbers here, which is the power calculation. And VAS no longer requires justification of the animal numbers, but it needs to be in the research strategy section. B, transplant about the analysis plan. So you need to have the clearly specify the statistical analysis plan in the scientific grant applications. For the biological variable, you probably want to specify sex and age, weight of the animal that you want to treat. That way it's very reproducible, and underlying healthy conditions and types of strains, vendors, source suppliers, and housing conditions. For those of you guys who's working with the mouses, right? The mouse living in the bottom of the cage versus top of the cage, do you think there will be differences in the conditions? And the one maybe facing the doorways versus the inside, or like lights versus the other. Those are the different variations that can come in. That's why you need to randomize the animals to see whether these things affect or not. And types of the studies, whether you're doing the tissue, or cells, or samples from the animals, veterinary animals or humans, and whether you're considering both genders as well. And so unless you have a strong justification for why you're doing only one sex study, you do need to include both genders in the animal study. And when you when you report the study, data should be disaggregated, whether study was statistically powered to detect the sex differences or not, and report descriptive statistics for males and females for the later future study. And studies that control for sex in multivariate analysis should also report sex-specific results to see whether there's a difference by the gender as well. And will I have to double my sample size if I have to include both genders? No, it depends on the outcome that you are measuring. That makes the difference, not the gender. So you can divide the animal number into each gender and see how the things go, but you don't have to double the sample size. It all depends on the outcome that you are measuring. And this is an example from the NIH, the reproducibility website. It's a well-written the grant proposal. And here's an example. Male and female mouse will be randomly allocated to experimental group at age 3 months. I mean, randomization is included as well as the exact age of the mouse. And at this age, the accumulation of CUG, repeat RNA, sequestration of MBNL1, splicing defects, and different conditions. And so it tells about what happens at the age 3 months. And the compounds will be administered at 3 doses, 25%, 50%, 100% of the MTD for 4 weeks. For how long compared to the vehicle treated to the control group? So 4 groups information and specific doses are spelled out. And IP administration will be used unless biodistribution of the study indicates a clear preference for the IV route. So method of the injection or method of the delivery of the medication is clearly specified. And group size of n equal to 10, 5 male, 5 female will produce 90% power to detect a 22% reduction on the CUG repeat RNA in quadriceps muscle by using the PCR. So it spells out how it is going to measure it, right? And what are the outcomes that we're looking at. And then like the treatment assignment will be blinded. Blinding is indicated. And then they also talk about their experience of doing this kind of research. So this is very good example. There are 2 additional examples available at this website. I think you can follow for your grant proposal as well. So Dr. Carter talked about statistics is unique among academic disciplines in that statistical thought is needed at every stage of virtually all research investigation, including planning the study, selecting the sample. I mean, if I get to know the study better, I'll be able to, statistician can help better to design the study, doing the better sampling, and managing the data as well as interpreting the results. Another way of looking at the statistics is there is a deterministic event as well as a random event. Deterministic event is everything is fixed. If I draw money from ATM, right, I will know how much it will be left. That's deterministic example of deterministic event. Whereas the random event is where all the experiment, there may be a random error that may happening in the experimental results. One example of the random event is, let's say we have like 100 of us in the auditorium, and if everybody tosses a fair coin, what is the chance of seeing the head? Fair coin, we get a 50% of chances of seeing the head and 50% of chance of seeing the tail. Is that means we're going to see exactly 50 head and 50 tail? It will not happen, right? It's a random phenomenon. So the worst case scenario, we may see 1 head. On the other spectrum, we may see 99 head, right? So having a 1 head coming out or 99 head coming out, do you want to claim a victory based on it? It all depends on the sample size and how you want to interpret it. That's when the statistics comes and help you with the probability theory. Based on the probability distribution, you can make an inference back to the population that you're studying. So steps in using statistics in grants, you want to consult with the statistician early enough so I can understand what you guys are trying to study and ask questions. It's a two-way street communication, and identify the questions and identify the type of study you're conducting and decide on the appropriate endpoints and decide on the appropriate analysis and help you with the power and data management plan too. And if you're conducting a study, like let's say you're conducting a study on the breast cancer patient with the stage 2, and overall survival happened to be 5 years, and you are trying to study R21, and duration of R21 is 2 years, and overall survival, let's say it's 5 years. So can this be feasible to conduct a study within 2-year time frame? That's not, but that happens with a lot of junior investigators that I interact. So what should we do? Instead of using the overall survival, we probably need to pick a surrogate marker or surrogate endpoint, which could be a tumor response. as an endpoint instead of using the overall survival, right? So those are the communications that needs to go between back and forth with the statistician and the clinician. And I added a little clip here. This happens in a lot of my cases here. Happens here at UAB too. It's the communication is key in the collaborative research. And let's watch this YouTube video. Some of you may have seen it, but it's never enough to emphasize the importance of the communication.
I have a study, and I need to know how many patients I need. I think I only need 3 patients.
Okay, what kind of study is it?
I'm doing a lab study. Can I just use 3 patients?
That depends. What are you trying to show?
We always use 3 patients.
Okay, what are you trying to show?
I'm trying to show that drug A and drug B are better than only drug A.
How are you measuring if it is better?
We are looking at apoptosis. Can we just use 3 patients?
What kind of measure of apoptosis? Do you know how variable that measure is?
We always use 3 patients. That is what we have published before.
3 patients per group or in total?
Yes.
Okay, let's try something different. What type of measure are you using? And is this an in vitro study? An in vivo mouse study?
It is an in vitro study of patient samples, and we are looking at measures of apoptosis after treatment with drug A and drug B versus drug A and control. And drug B alone.
Oh, how many groups are you analyzing altogether then?
My grant is due tomorrow. I just need to know if I can just use 3 patients per group.
Okay, well, you probably should have come to me earlier for help with this.
All I need you to do is tell me that 3 patients are okay.
So, it is not that easy. Your marker for apoptosis may have a large amount of variability associated with it that could make it difficult to ascertain differences based only on 3 patients per group. It also depends on what kind of outcomes you are looking at. Are you looking at normalized changes in expression levels from baseline? 3 patients may not be sufficient to detect differences in these expression levels between the different treatment groups.
I do not understand.
I'm saying that you may need more than 3 patients per group, but that I need more information to determine that.
The serine protease granzyme B is made in cytotoxic lymphocytes, where it is inactive due to the low pH of the storage granules. It is measured immediately upon delivery into target cell cytoplasms, where the pH is neutral. This is followed by cleavage of procaspase 3 by granzyme B in the target cell cytoplasm and the subsequent induction of apoptosis. All proteolytic events can be quantitated at the live single-cell level by flow cytometry and imaged by confocal microscopy.
I understand very little of what you just said. Maybe we should talk more about this.
I just need to know if 3 patients are enough. My grant is due tomorrow.
We will need to meet to better understand this before we can determine the appropriate sample size.
I can put you on my grant for 0.5% FTE. You can design and analyze the data later.
Do you really think that is enough to do the work?
Well, it may be less if it is awarded and the budget is cut.
Please go away.
So, I mean, that's the real, scenario that happens here at UAB too. So my key point is it's a two-way street. I need to understand the lingo that you guys are using. If you're receptive, it just takes time. If I'm used to communicating with the investigator, it doesn't take long. But if it is like if you're someone coming up at Friday afternoon and my grant is due on Monday, that happens here too. And I can come up with something, but that's not the best reflection of science that's in the grant. When we were preparing the CounterACT grant, we had a lot of communication with the lab personnel and going back and forth, who's doing the actual experiment, like asking these questions, that questions, and a lot of communication back and forth, and that resulted in a good grant application that got funded as well. And And the PIs were very receptive about the asking for, can we have a meeting? I mean, let's talk about this. And that's very crucial, I mean, to have a two-way communication. Otherwise, being a statistician, I work with all different kind of data that's coming in. And if you come in like Friday and doing this like totally different language and talking to me, like it goes above my head. I mean, you're the content expert. Me, I'm not the content expert. I mean, like I get a like simple email sentence or paragraph. Yeah, here's what you need. I mean, can you do a sample size calculation? No, I mean, I need a lot more information to figure out. I mean, like the YouTube video that you watch. A lot of communication needs to happen to produce a good, reproducible scientific grant application as well as a research. This is a slide that I borrowed from the Dr. Liu, who's at the Weinstein Institute Cancer Research, and determining the treatment effect using longitudinal tumor growth data from a mouse model. So typically what happens is when you do the tumor growth data from a mouse model, and here's the experimental procedure. They randomly assign the mouse to 4 groups, 10 mouse per each, and then they measure the tumor volume over the time. And they look at they measure the tumor at the baseline, day 4, day 7, day 9, day 12, and day 14. And usually what you do, you look at the ANOVA at each time point, right? That's the typical practice in the lab. That's what I heard. And so the ANOVA only looks at the fold changes at the last time point. And if you look at it, the results are, it looks like there is a difference in the group, but because you get to do the multiple comparisons, you got to apply the bumpering adjustment, which makes it the comparison a little bit more conservative. If you look at it, control versus the GW2580 is not significant, and control versus the PLX4720 is significant. Combination is significant, but there is no difference in the control versus GW2580. So, this investigator went and talked to my colleague, and the approach that they took is looking at the velocity, trend of the tumor growth, right? Speed of the tumor growth. That's what they used to analyze the data. Instead of using the fixed ANOVA model, they used the fixed mixed effect model to look at the trends of the thing. And if you look at it, the combination group, the tumor volume, the green one volume stayed flat, and PLX, which did not show a statistical significance by just measuring the tumor volume, but if you look at the trend, speed, right, there are significant difference between the trend between combination and PLX. So it depends on what the outcome you're looking at, right? This is another way of looking at the data and analyzing the data. So if you come to the statistics, you can utilize all the data that you have instead of just looking at each time point separately. You need to utilize all the data that you have. That will give you more power to conduct a proper statistical analysis. So communication is another key, on these as well. So, like in the statistics and rigor and reproducibility criteria, you need to identify your study questions, and it usually comes in a hypothesis, which is defined as an assertion, the subject to verification or proof, as a proposition stated as a basis for argument or reasoning. In most of the cases, we get to have a null and alternative or researcher's hypothesis, and we want to prove or reject the null hypothesis. And based on your the study, you need to define which type of study that you will be conducting. It could be observational study or comparison study. It could be a descriptive study or case scenario or analytical study, cohort study going, exposure to outcome, outcome to exposure, or cross-sectional association study, or randomized experimental study. Randomized experimental study is the gold standard, which happens a lot in the lab environment. But if you go to the human studies, case control study or exposure to outcome or outcome to exposure may be the case. You cannot expose the the cancer drug to a human at the healthy population, right? So it's not it's not the best. It's not the right study design. Depends on types of the study, different types of the study design can be developed. And as we discussed earlier, I mean, choosing a proper endpoint depends on your resource that you have it available and the duration of the study as well. When you conduct a study, you got to think about like how many you can enroll to a study if you're doing a clinical trial. If you can enroll, let's say, 20 patients per year, and your sample size requirement is 100, you may not be able to conduct the study as a single site. So you may have to go to the multi-site study. And then outcome, as we discussed earlier, if you cannot measure that outcome, overall survival within 5 years, you may have to search for the different outcome, surrogate outcome as well. And when you're choosing the outcome measurement, it must be appropriate to the study question being addressed and must be accurate and approximate the measurement of the phenomenon. It should be complete. I mean, and then it should not be influenced by the process of the observation. If there is a missing values, you have to think about how you're going to treat the missing value and how you're going to do address the attrition as well. So we talked about the random sample earlier. I mean, tossing a coin example. Basically, let's say, I mean, you do an experiment. That's a random phenomenon. You want to take it back to the whole population that you want to influence it back to. Population is very large, perhaps an infinite number. How do we calculate the estimate from the sample? You guys do like mean, median, and survivor, median survivor, and proportion. Those are the estimates. And how can we say about the accuracy of this estimate? We do capture the estimate based on the confidence interval. And how can we make inference about a population from the sample? That's based on the probability inferential theory. And how confident are we about the correctness of those inferences? So we're going to look at the one example like tossing a coin. A researcher is studying a treatment for a disease that usually kills half of its victims, so about 50%. And he treats 10 patients with an experimental treatment, and 7 survives and 3 dies. So then 70% based on the sample size of 10. Is this a good enough evidence to move forward? So logical basis is like, it is not likely that something happens about 50% of the time. In the long run, we'll have 7 or more. But I mean, you may be guessing 7, that may be a good indication. But decision problem, right? If you are like FDA committee, serving on the committee, will it give you enough evidence to prove whether this drug is working? Or if you are a director of research, you have to invest like millions of dollars, 7 out of 10, does it give you enough signal to do an investment? Maybe not. So this is based on the probability. 7 out of 10, 7 heads out of the 10, 7 alive out of 10 is equal to the probability is equal to 0.17188. Typical p-value that we used in the study is 0.05, right? So that does not meet. I mean, that basically what it's telling is the chances of 70% is not much different from the 50%. So if you want to move forward, you probably want to see 9 out of 10 of the survivals. To move forward based on the statistical p-values. And confidence interval based on the 10 observation goes from 0.3475 to 0.9333. What is the confidence interval tells? That basically tells about the confidence of this estimate. So what happens is if we do this experiment with the 10 observation or 10 patients over and over and over again, one occasion could be as low as 0.3475. Just like tossing a coin example, if you do it like 100 times, one observation, one experiment, we may end up in the 0.9333. So those are the ranges. We happen to see 70% proportion, But if you do an experiment next time, it can go as low as a 0.3475 and as high as 0.9333. If we want to have a smaller confidence, then we need to increase the sample size. If we have a 70 survivor out of 100 patients, then the confidence interval gets narrowed. So it goes from 0.6002 to 0.7876. That's why we need to do a proper sample size calculation. You can do a precision calculation for this based on the sample size. So it's very important to have a proper sample size and calculations to move forward. And moving into the power and sample size calculation for the study, and there's different types of the things that gets to be mentioned in the grant application. Type 1 error and type 2 error and power. Type 1 error is alpha level, and type 2 error is a beta, and power is equal to 1 minus beta. And these are translated into, in terms of the clinical justice system, let's say your null hypothesis is dependent is innocent, and alternative hypothesis is dependent is guilty. Type 1 error can be translated as a probability of finding the dependent guilty when the dependent is really innocent. Type 2 error is probability of finding the dependent not guilty when they are indeed guilty. Power is probability of finding a person guilty when they are really guilty. That's what we want to prove. That's the power of the study. So some of the important things to consider in the sample size calculation is what is the desired effect, detectable difference that you want to achieve at the end of the experiment. It needs to be statistically significant as well as clinically significant. Just having a large sample size, you may find the statistically significant. So one example could be, let's say you develop a wonder drug that reduces the blood pressure, SPP, of 5 units. And it happened to be statistically significant. P-value is equal to 0.0001. But the actual mean value of the SPP was dropped from 180 to 175. It is statistically significant, but dropping of the BP from 180 to 175, 5 units, is not clinically significant at all. So those are the other things that you have to consider when you do a power and sample size justification for the primary outcome of your study. And what previous information do you have about the variability? It usually comes from the previous experiment. If you do not have a source to the previous experiment study, that variability may come from the published manuscript. And do you have a limitation on the sample size? As we discussed earlier about the clinical trials, right? I mean, if it is the clinical trial, how many can you afford based on your budget and resources and time duration of the study? And you can look at the other outcome as an exploratory outcome. You can do a proper sample size, just primary outcome as well as sample size justification based on the different outcome. And what are the estimate about, what about the dropout? If you're doing a clinical trial, what is the estimated dropout rate? Are you expecting a 10% dropout, 20% dropout? At the end of the study, how many do you need? And also, what are the special considerations that you have to give? Are you trying to conduct a group randomized trial, a cross trial, and so on? Here is one example that I had to convince the cancer center investigator. Scenario 1 is the investigator wanted to have a head-to-head comparison on the 2 treatment arms, comparing the toxicity of the two drugs. However, the budget that he had from the company only allowed to assess up to 50 patients. So, but he could not afford the 78 patients. Budget was not allowing to look at and then he was also interested in looking at the progression-free survival as well as the overall survival. So instead of having a 2-arm comparison study, I suggested to do a 2-arm study, but it's not comparing, but do the comparison within the arm with the historical control. So you can also look at the toxicity within each arm, not just do the comparison across. But we also were able to get look at the secondary endpoint, which that he wants to get it for moving forward to a different study, which was the progression-free survival as well as the overall survival. So depends on the outcome that you want to measure and the resource that you have available, different types of the study design as well as sample size justification can be provided. The key components of the power section in the grant application is description of your assumptions. What are you assuming, and how many, what are the variability and dropouts and so on. And then basis for your desired detectable differences, which should come from clinical as well as a practical difference. And then it also must be clear and reproducible. And calculation should account for the potential dropout. And if possible, hypothesis should be hypothesis-specific calculation should be provided. Moving on to the, there are many different types of statistical analysis available, and then new types of techniques are developed in the statistical world. So having a conversation and what you want to achieve with the statistician, what you want to really help for you to use the cutting-edge statistical techniques that's available out there. And this is another example from the cancer center. So what happened was those of you guys who's in the clinic, disease-free survival rate would be less than overall survival, and greater than overall survival rate. Disease-free survival cannot be more than overall survival. But he learned how to use the black box statistical software for the SPSS and click, and this is coming out. What's going on? With that, I cannot explain what's really going on. I could sense what may be going on, but when you conduct a statistical data analysis, there is a thing called assumptions that you have to check for each data that you're looking at. Normality assumption may be one thing. And then also when you conduct a survival data analysis, you gotta look at the missing data. If the data is more than 30% missing, you really have to think about the validity of this survival analysis. So this is the graph that he produced at this end. And what happened was to assess the disease-free survival, you need to know the relapse date information. If you look at the relapse date information here, most of them are missing. Then your survival analysis may not be valid because of the missing assumption. Missing is the assumption. You can't have more than 30% of the data missing and try to conduct the proper data analysis. So key components of the grant statistical section is you need to do a proper description of the study that you're doing and understanding of the nature of your data, and clearly define the outcome, and appropriate and state-of-the-art statistical analysis that's available, and any special considerations and multiple testing or multiplicity issues. If you're doing 10 different outcomes, you probably need to do a proper p-value, inflation of p-value adjustment, and hypothesis-specific analysis, and missing value plans, and attrition analysis plan that needs to be added as well. So these are the most common mistakes that we get to see in the grant application. Power calculation is not reproducible based on the information that has been provided, and power calculation does not contain the prior information. Statistical methods proposed are not appropriate based on the power and sample size calculation that's provided. And sometimes statistical method is too general, it's not specific, it's like coming off from the textbook, and that's a big no-no. And lack of detail regarding data management and quality control, and too little report like you've seen on the video.